Well, thanks very much. I think this is a really fabulous thing you're doing, this Microbiome Virtual International Forum. It sounds like a great way to use what we discovered, as you said, during the pandemic, different ways of communicating. And I'm incredibly honored that you chose me to kick it off today. And so I'd like to thank Levi and Nicola and the steering committee for bringing me here today. And I'm excited to tell you about what we've been doing, and a lot of what I'm going to tell you today is also unpublished work, but we're really excited to be sharing more broadly at the moment. So I don't think the microbiome needs much of an introduction, so just let me begin by saying that we know now from a couple decades of research that microbiomes differ between people. One of the ways they differ is compositionally, where if you look at microbiomes of different people and you look at the relative abundance of the types of microbes that make them up, you will notice quite often that there's differences in the relative abundances of different microbes. And so a lot of the field and a lot of the ecological research has been to try to understand what drives these kinds of differences between people. And a lot of the work has centered around the role of the community we come from, the diet that we eat, the lifestyle that we have, our health, and so on, to try to explain the patterns that build up over a lifetime as we're colonized early in life, and the diversity builds up over time, such that by adulthood we have our own unique microbiome, but that also shares similarities with other people. And what my lab is focused on, aside from the environmental factors that can shape the microbiome that we consider background noise, we've been trying to get a handle on whether some of the differences that you can see between people can be attributed to differences in their genetic makeup. So within a population, if you have a bunch of people that you've profiled, if you know their genotypes, can you then explain some of the variation between people in compositional data if you know their genotypes? So this is the strongest signal that's come out of this kind of work. It's come out of our work, it's come out of other people's work. This is by far the strongest signal of the effect of genotype on the microbiome. And it has to do with the LCT locus, or the gene that encodes lactase, which is the enzyme that breaks down the milk sugar lactose. And so people are either lactase persistent or not as they go into adulthood. And what we see is, for the lactase non-persistent genotype that have a low expression of lactase, if they're drinking milk, if they're ingesting lactose, there will be a greater proportion of bifidobacteria in their gut bacteria if you compare them to people who express a lot of lactase, who continue to make lactase into adulthood, the lactase persistent genotype. So in these people, you will find relatively lower levels of bifidobacterium. So this is a straightforward story, it seems, of lactose coming in and either being delivered straight to the bifidobacteria that thrive on it if you don't produce lactase, or if you do produce lactase, you will access the sugar yourself, take up the byproducts, and there's less for the bifidobacteria. And so relatively, you'll have fewer of those. So this is something that occurs at the genus level. We have looked more precisely to try to figure out maybe the species or strain level patterns here, but no, it looks to be it's really all of the bifidobacteria are enriched together and proportionally. And what's interesting about this example is that lactase persistence, the development of lactase persistent, is also one of the strongest signatures of selection on human populations worldwide. So lactase persistence arose during the Neolithic period, when people domesticated animals and started using milk in their diet. And there's a couple centers where it arose independently. So it happened in Europe, it happened in North Africa, it happened in a few different places. The genetics are different in each place. But it is one of the strongest signals that we have in our populations today that humans went through some sort of bottleneck in a few different places, such that being able to metabolize milk sugar into adulthood gave them an advantage, and these genotypes spread. So you can see that there's quite a lot in Europe, as you probably know, and then there's some other places in the world where this genotype is more abundant than you'd expect. And the original state, of course, is lactase non-persistent. So this happened in response to a big change in diet that was culturally driven, and it has led now to a big effect on the microbiome that we can still see, that we see today. So lactase persistence is just one of the genetic adaptations that humans made as we moved around the world and encountered new environments. And this is a nice summary of some of these genetic adaptations that Sarah Tishkoff's group put together a few years ago, published in Science. So each one of these represents genetic adaptations to environments that people ran into and adapted to. So drinking milk's one of them, adaptations to cold, adaptations to altitude, adaptations to different pathogens that people ran into. Another one is adaptation to starchy food, and this would be this AMY1 here. And this is another one that a couple of years ago now, my group investigated to see if this was also, and this is related to diet, if this was also reflected in the microbiome, the differences that people have at this locus. And so what happens at the AMY1 locus, and this is the gene that encodes salivary amylase. Salivary amylase begins the process of starch digestion in the mouth. And humans have copy number variation of this gene that ranges from about 2 to 20. And the more copies you have, the more salivary amylase you produce, and the faster you'll start degrading starchy foods in your mouth. So a couple of years ago, this is Angela Poole's work. She's now faculty at Cornell. She took a look at people that varied in copy number, in gene copy number, high and low, the same kind of idea that I just told you about with lactase. So if you have a low copy number, you will have less of this amylase accessible starch being digested early on, and all of these starches can reach the gut microbiota. If you have high copy number, what we believe is happening is you're accessing some of these starches that are then depleted from the chyme, so that by the time the chyme reaches the large intestine, what might be left for the gut microbiota is more resistant starches, and those are accessed by members of the Ruminococcus genus, for instance. And so in high copy number people, we see an elevated percentage of Ruminococcus compared to the low copy number of people for the AMY1 locus. So here's another example that's also based on diet. We know that humans, and this is probably predates the Neolithic, but it was probably enhanced during the Neolithic, really started to add more and more starches to their diet. And this might have been one of the consequences. Now, nowadays, there's this copy number variation in every population we look at. So we have this standing variation, and it's one of the things that could be on a daily basis, if you will, shaping the relative contribution of these different microbiota to the overall microbiome. So Taiichi Suzuki in the lab was thinking about this recently, about how you have genetic adaptation of microbiota to different parts of the world and to different environments that people might have encountered that are well documented by human geneticists. And if you go into the literature, you can find for all of these different selective agents, you can also find evidence that the microbiota can also vary according to these selective agents or these traits in humans. And so this might be giving us a map and a way of thinking about why it is that genetic diversity in people could be reflected in the microbiome. And we've only looked at a couple of these in detail, but this is giving us a roadmap, if you will, of others that we might be able to look at to see just how the microbiome reflects our genetic diversity, and then to think further about how it might have actually played a role in our genetic adaptation to new environments. But there's another kind of variation that we can see, and this has come out of Nicola Segata's lab particularly, because they developed the tools to look at this and have seen some of these patterns most prominently. And that's that if you look in microbiomes of different people around the world and you look at within species, you'll see that there's different dominant strains. So now, if you only look at Prevotella copri, for instance, in different parts of the world, you'll see strain variation and dominant strains in different populations around the world. And that, again, is something that you could attribute to differences in environment and lifestyle. And these are definitely going to be involved in that. But we've been thinking about what our evolutionary history could have done to contribute also to these kinds of patterns. And this is something that Andy Mueller at Cornell and Howard Ochman were thinking about at a larger evolutionary scale. When you think about the evolution of our species from our hominid ancestors that gave rise to our closest relatives, the gorillas, the humans, well, okay, we're the humans, the bonobos, and the chimps. This is our tree of the hominids. And using single gene markers, in this case, gyrase B, they were able to see that particular types of gut bacteria, here's bifidobacterium, gave these strain patterns, so these species patterns that match very well the host phylogeny. So here's gorilla's basal. Here's a couple different bifido strains in humans. Here's the bonobos and the chimps, and it looks like here there was another jumping, probably, of a strain into a gorilla and a nucleate here. And so what and they showed this for some bifidobacteria, from Bacteroides. They couldn't show it for everything because not everything has gyrase B. But there were definitely these species of bacteria that have co-speciated with hominid species. So this implies that these bacteria are being vertically transmitted over time and faithfully within these lineages, such that even as we diverge as species, the bifidobacteria, for instance, diverge as species as well. So this implies very faithful vertical transmission over thousands, tens of thousands of generations. And so if you think about this and extrapolate forward, could this continue to have happened within humans as we spread around the world, and can we see any sign of that? We came out of Africa, we spread across Eurasia, the land bridge into the Americas, down into the Americas, and most recently into Australia and Micronesia and places. So you can imagine that if we've had very faithfully transmitted strains following populations, that you could see differences in strains that might reflect these large scale patterns. And in fact, this is not a new idea. You probably know the story of Helicobacter pylori, which is the stomach pathogen. And almost 20 years ago already, David Falouche and colleagues, using isolates of Helicobacter pylori that were collected around the world, showed that the strain distribution was consistent with patterns of human migration out of Africa. So the question that we've had is this true for other species of gut bacteria? And is there a way, can we do this now without having to make isolate collections? Because I think you all know that that's a big task. So can we find other gut microbes that have co-diversified with humans, and can we do this using metagenomes? Because Nicola Segata has given us these fabulous tools to pull strains out of metagenomes. And so what we could do, the idea is that you take the strains out of metagenomes, and for every species that you can do it for, you build a tree. And then you compare that tree to your host tree. So in this case, a tree from different human populations. And you're going to ask then if the bacterial tree matches the host tree, just like bifidobacteria matching the hominid tree. So this would be indication of codiversification, this kind of matching A frequent exchange between hosts would give you a more jumbled pattern, and then we might see something in between. So we would expect bacteria to fall on a gradient between frequent exchange and codiversified. And can we find bacteria that fall into these categories? So now I'm going to tell you about how we've approached this. And first, I want to introduce the team. So this was a big effort and involved a lot of people from my own group, and here they are. So Taiichi Suzuki is a postdoc who has led the co-phylogeny analysis. Liam Fitzstevens is a graduate student who, along with Victor Schmidt and Mirabeau Bong, did a lot of the sampling that you're going to see, and then he's also been focusing on strain sharing between individuals. Hage Inav has also been focusing on strain sharing using synteny as a way of comparing strains. We have Nick Youngblood, who's a group leader here and has provided a tremendous amount of bioinformatics support to the team and does things like, whips out mags, which I'll show you in a minute. And Kelsey Hughes is another postdoc who has recently been coming up with clever extra ideas for the project. And then these are our partners. So first of all, Peter Kremsner, who leads the tropical medicine clinic here in Tübingen, has facilitated sampling both in Vietnam with his partners and in Gabon with his partners, shown here. And on top of that, we've been able to throw in data that we collected previously, working with Tim Spector from the UK. Those of you familiar with our previous work, this is the Twins UK data. And then something that was really terrific and really encouraging about sharing science is I've given this talk a couple of times now, and as it's been developing, and Laure Sigurel from the CNRS in Paris gave me a call and said, hey, we have some data that would fit your study. And she developed those data along with Rand Blackman from University of Minnesota. And so some of the data I was showing you was recently contributed by them. So these are the data that we generated. We went to Vietnam, we went to Gabon, and then we went down the road here in our local town in Tübingen, Germany, and we sampled metagenomes from mothers and their babies. We didn't get babies for all the mothers, but basically these are women, and we were, we really wanted their babies as well. So there's a lot of kids under 1 in this study as well. And so these are the kinds of numbers we're looking at, and we generated genotype data for the hosts. So as I said, we also added our UK samples from the Twins UK cohort and Cameroon samples through Ran and Law. So in all, we have 747 adults that are included in this analysis, and we have fecal metagenomes from them, and about a little over half the children. So we don't have children from the UK and Cameroon. We have children from Germany, Gabon, and Vietnam. So this is the host phylogeny that Taiichi has put together using SNP data. So close to 40,000 SNPs gives you this kind of tree. And you can see it's colored by these 3 major groups. You've got Africa, Asia, and Europe. So if you look within Africa, you can see Taiichi's labeled the Cameroonian samples, the Gabonese samples, and these here, interestingly, are forest people, 2 different tribes. You can see that they're a bit more genetically distinct from the rest of the people of Gabon and Cameroon, but they are each other's closest relatives. So we have, as you'd expect from a human phylogeny like this, Africa basal, followed by Asia and Europe. Germany and UK people are indistinguishable at this level of analysis. So now some of the microbes. So we were able to run this analysis for about 60 species, 57 in total. And here are some of the ones that show the most striking patterns by eye. So for instance, Collinsella aerofaciens shows you Africa first, followed by Asia, followed by Europe. Here's Rosburia hominis. There's a bit fewer of them. As you can tell, the branching pattern is not so dense, but you've got Africa, Asia, Europe, and then Asia. So we have these bursts of strains. So particularly Prevotella copri, this has been described before. But you could see that each time we have, generally we have Africa basal. Here's your bacterium with a big strain from Africa, some mixed strains here, and then you can see, so they're varied, but you have these pretty striking population level strains. And the question for us now is, how well does this match not just a 3-part tree, but also within populations? Can we see evidence of cophylogeny? I'd like to point out the 2 tribes of forest-dwelling people are here and here together. And to give you an example of one that doesn't show a pattern at all, so this would be very mixed up. You've got Parabacteroides dististonis here, and then a Bacteroides. So these would be examples of complete mixing, where we really don't see any of this strong population level patterning, and absolutely no sign of cophylogeny when we run the statistics. So this is the kind of statistics that Taichi's been running. The idea is, how well do these trees match? And do they match better than they match by chance? When you take for each one of these 57 and you ask, here's my bacterial tree, here's my host tree, how well do they match? There's different statistical tools that can be used to do this. So Taichi's run 3 here, and now he's sorted the list by the most conservative test first, and sort of by the he's ranked them by the ones that give the highest signal of cophylogeny to the lowest. So we already see some top taxa up here. All this yellow is showing you that these ones at the top are giving us significant cophylogeny across multiple tests. And here, highlighted in blue are the firmicutes, and highlighted in red are the Bacteroides, and it really looks like the firmicutes are at the top of the list. It's significant, actually, as well. So there's something about these firmicutes that are pushing them towards the being more cophylogenetic with humans compared to the others. Another thing that's interesting that Kelsey pointed out is that if you take on the x-axis this evidence of cophylogeny, so how many tests are they passing versus genome size, we see that the more cophylogenetic you are, the smaller the genome. And this is something that might be expected if these taxa are really starting to become quite symbiotic with us. The other thing that is important to do here is to run these tests within populations as well. So within populations, so if you only look within Africa, within Asia, within Europe, it's much less, it's actually more difficult to get significance, because now you have a smaller number of people included in the tests. But you can see that we're also picking up signals of cophylogeny within the populations. And so that really does support the fact that that it's not just, say, diet or environmental factors that are causing these population-specific clades to emerge within these species. But it's tougher to argue that it's just diet when you're looking within a population. Finally, so I forgot to mention that we use strain flan here to make, thank you, Nicola, for strain flan. We use strain flan to produce the trees that went into these, analyses. We've also been able, but there's less power to do this, pull out metagenomically assembled genomes, or MAGs, and make trees out of those. And so this is an independent approach. We can't get good enough MAG coverage on all of these to be able to do these analyses, but we can on, well, a good half, and you can see that they're passing the cophylogeny tests in the same way. So it's the top ones again, and so that's a nice separate test that we can run. So another thing, I pointed out on the trees that you see Africa basal a lot in these strain bursts within the species wheels, and you can now ask if there's been more strain transfer events from Africa to the other populations than the other way around, for instance. And so this is a test for that. using the number of significant tests as evidence for cophylogeny and counting the number of transfer events, you can see that out of Africa is definitely something that the strains with cophylogeny are exhibiting, not out of Asia and not out of Europe. So this is evidence similar to what we see in H. pylori, that we have an out of Africa pattern here. We wanted to know how general this was. So can we expand this to more data? Because this doesn't actually need the host genotype data. So we can go into public metagenomes and expand the data set and ask about out of Africa. So Taichi took over 1,200 human fecal metagenomes from 19 countries, and also primate fecal metagenomes, because we can use these to essentially, these should be basal to all the humans now. And he did this for the top taxa, and he did this for the bottom taxa. So now we're just comparing the most cophylogenetic with the least cophylogenetic. And here are examples of the top taxa using the public metagenomes. And so now we have a lot more countries represented. It gets more complex if you want to look at the details of the countries that are shown in the ring. But here's, for instance, Prevotella copri. And we have Africa basal here. And then at the next burst of strains, you have a primate basal. For all of the others, the primate strain is always basal, as you can see from these. And this is an example of the opposite. These would be the bottom 7, and the primate ones are all over the place. Everything's all mixed up. There's no Africa basal here. So to show you this a bit more quantified, this was our original 5 populations, top 10 compared to bottom 10. Do we see transfer events, Africa to rest? And the top 10 definitely have more out of Africa transfers than the bottom 10. Here's Asia. Here's a bit of a trend with Europe to the rest for the bottom 10, which is kind of interesting. If this would hold out, this means that things that transfer, that don't transfer vertically, that transfer from, fecal oral route, are spreading from Europe out. So here it is with the full set now with 19 populations. Out of Africa significant. Now we have America to the rest, which isn't Asia to the rest, and the trends with Europe to the rest going the other way, where the bottom 10 tend to be spreading to the others more. So this recapitulates what we've seen in our own populations, that we have Africa to other places in our top cophylogeny scorers. So I told you that we had kids in the data set. I'm not calling them infants because some of them are a little bit older, and the average is about 7 months. And as you know, babies, infants, kids, young children have different metagenome, different microbiomes compared to adults. And so it's not surprising that we picked up other types of taxa, such as bifidobacteria, showing us patterns of cophylogeny. So here are some of the trees. And it's not too surprising also that it would be bifidobacteria, because these have been shown by others to transfer between mothers and their own kids. And we've also seen these longer term patterns in the hominids. And so here we are again. These are showing patterns of codiversification. The one that we could compare between adults and children was Prevotella copri. And so you see, there's a lot more of it in adults. We did have fewer children, but it's one of the only taxa that we could do this for both adults and children. And it is showing us cophylogeny in both adults and children. So here are the stats for the kids. Here are the top are bifidobacteria. We also see the patterns within Africa, within Asia, not so much within Europe, but Europe has less genetic diversity. It's less interesting in that sense. So with all populations and also within populations, we're seeing these signals. Bifidobacteria popping up as some of the top of the list, and Prevotella copri again. We see it in kids, we see it in adults, we see it within countries. what we wanted to know then, if we see these patterns of cophylogeny are implying that there's been long-term vertical transmission over hundreds of generations. So we should be able to see it now in the generation that we have now. And so we've used InStrain, thanks again, Nicola, for the tool, and a Synteny-based strain comparison tool that Hage Enav in the lab developed in order to compare mother-infant strain sharing to unrelated person strain sharing. And so this is showing you that we basically have the same taxa that we can look at using both methods. And again, a ton of Prevotella shared between mothers and their kids. And so we're seeing the exact strains, as has been noted by others, but it's a nice, it's consistent that if you see it over long term, you see it over the short term as well. We're watching the vertical transmission happen. So this is a first pass at a list of gut taxa that are showing cophylogeny with human hosts. I think the more we expand the analysis, expand the data set, I would assume that we're going to see more and more support for this, and maybe the list growing. The patterns that we're seeing, such as out of Africa, these are supported by our additional populations. We also see immediate strain sharing now, not only this long-term patterning. So all of this is telling us that this sharing of microbes that happens within families, within tight groups, if you extrapolate to migration patterns and you look at populations now, this is one factor that can explain why you see different dominant strains in different places. So it's entirely consistent with diet and other things, helping shape those strains, helping maintain those strains. It's just that we also now have this additional dimension, which is that people also could have carried them there from the beginning. So we have gut microbes that are specific to us and some that we believe are specific to us at the population level. And now we can start to think, or this is where we're going to be going next, is what is special about them. So there's microbes that are following the oral fecal route that are widely spread, things at the bottom of the list. These include more Bacteroides, they have larger genomes. It would make sense that they're better at surviving outside the host. These are things we could test. We could look for what kind of genes are supporting this more complex lifestyle. Maybe they're more likely to be found in other mammals, and maybe they have a different kind of role in the host. And then we have the parent-to-kid route. So H. pylori is an absolutely classic example of this. You only pass it through basically vomiting on your children. It only survives 4 seconds outside the host. Others that we're finding, Prevotella rosebura, it might not be that extreme, but it must be pretty tight. So do they have poor survival outside the host in comparison to these? How do they differ between the populations, and what does that say about their role? Does it say anything about their role in health, and is that population specific? These are all the kinds of questions that'll be fun to think about going forward. So I'm going to end there and extend thanks to everyone in the group. The kind of work that we do is extremely collaborative, and so we have a lot of collaboration within the group. We have a lot of collaboration outside of the group. It's a really great community, both within and outside of the group. It's terrific to be able to use the kinds of tools that others develop and share. And if you would like to come and work with us on more of these questions, then please do get in touch.